use activity, because there was some that was very much surplus, and there was depletion of other minerals. Does that tell anything about how the soil after that?
So the question was, does the SAP analysis give us any clues or indications around what's happening with the soil biology? And the answer to that is sure, indirectly, sure, if the plant nutrient analysis is not looking very balanced, we could assume that there's some level of the soil biology that's not functioning very well. There's something, maybe that nutrient that's low, for example, the organism that's a specialist solubilizer of that nutrient might not be functioning very well. So it gives you a crude suggestion that something is not functioning down below, but it doesn't explicitly tell us what, or how to kind of address that. It just tells us that likely the biology is not functioning very well if there are imbalances like that. Yeah, so maybe more indirectly we could assume so, but it's not explicitly measuring any biological metric or anything like that. Yeah. Okay, so we're talking here about nitrogen metabolism, processing, utilizing the nitrogen, turning it into those useful molecules that the plants require. And this next slide, we're just really expanding on the same idea. We're just going to kind of explore this kind of step around here onwards, this kind of the latter half here. Through this slide here. So again, if the plant takes up, for example, nitrates, of course, those nitrates are back here. They need to be first converted into ammonium. If the plant takes up urea, that needs to be first converted into ammonium. And once we get that ammonium, it's this step. Ammonium is the inorganic form. We're going to turn that into an amino acid. Okay, so we're going to get some carbon and plug that in, attach that to that nitrogen and turn it into the organic form. So we have those amino acids, okay, organic nitrogen, nitrogen plus some carbon. Then the plant will stitch those amino acids together to form things called peptides, that's small chains of amino acids. And then again, it can stitch those together to form these larger, more complex proteins. So the point is here just to say that each step of that journey does cost energy for the plant. It has to build the specialist enzyme that will facilitate that reaction. And that kind of drains or costs the plant metabolic energy in order to facilitate each of these steps. So, of course, the shorter the steps, the more energy we save. The longer the steps, the more energy we drain. So it just opens up this point to say, well, what if we just started feeding plants those organic forms? What if we just started feeding them instead of the inorganic forms? What if we fed the plants amino acids and peptides as the dominant source of nitrogen? And then it's really easy for the plant to finish using those. And so this is called metabolic shortcutting, or we are shortcutting the metabolic pathway and delivering nitrogen in an already organic, closer to the end game form, which preserves the plant's energy so it can use that energy for more biomass growth, more production. So then really that's the essence of this idea of the nitrogen utilization efficiency. How much biomass can I produce per unit of nitrogen? Well, it depends what form of nitrogen you supply. And if you're supplying a lot of these amino peptides, you're going to be able to produce more biomass because the nitrate and the ammonium are draining more energy to get through that same process. Okay, so that's the essence of that idea. So the aminos open up this opportunity for very efficient use of nitrogen. And we're going to circle back with some various numbers and examples of this. So let's start building that picture. Here, this is an example of looking at the energetic cost or the metabolic cost to the plant in order to use these different forms of nitrogen. So I'll put some numbers behind this for you, what I was just saying. So the chemical form of nitrogen that is taken up, whether that is the inorganic form, such as nitrate, or the organic form, such as amino acids, this may influence the plant shoot and root growth and therefore influence the nitrogen use efficiency. The key point here is that the carbon cost or the energetic cost for the plant to turn those organic nitrogen forms and finish the job into those proteins. The energetic cost of that is lower than that of the inorganic forms. And that is particularly also because of the carbon content of the organic forms. And we're going to dive deeper into exploring this. So this carbon bonus, the embedded carbon that comes with the nitrogen in the organic form, that is what adds a big part of the efficiency gain also, and therefore makes it more beneficial for plants to make use of organic versus inorganic forms. So here's what we're looking at here. We're going to do some carbon economics. So what you're looking at here is a table that says, well, how much energy does it cost of the plant to process that nitrate versus ammonium versus these amino acids? So let's start with nitrate. So what this number is saying, how many grams of carbon, and think of carbon as energy, because that's energy, carbon to build the enzyme, to facilitate these reactions. What is the carbon cost for the plant to process 1 gram of nitrogen? How much carbon does it require to process 1 gram of nitrate? So you can see here that the plant requires 5.81 grams of carbon, 5.81 grams of energy, if we can say that, to process 1 gram of nitrate. But you can straight away see that to process 1 gram of ammonium, it's less energy. It's 4.32 grams of carbon to convert 1 gram of ammonium. Now, that's less. So ammonium is more efficient than nitrate. And that's obvious to see why, because nitrate is up that end, okay, but ammonium is one step closer. So, of course, it's going to need less energy to finish ammonium than it is nitrate because it's already closer. Okay, so that's why those numbers are different. In order to process that 1 gram, it's 5.3 versus, 5.8 versus 4.3. Now, both of those are inorganic, meaning has no carbon embedded. So therefore, there's no free carbon that they're taking up. They're taking up an inorganic form. So the overall net costs remain the same. Okay, so that's processing your inorganic forms. When we talk about these inorganic forms, and there's two here, these are two different amino acids, glutamine and arginine, they are a little bit different. Now, first thing is to say that, look, there is a carbon cost still associated with the plants acquiring those amino acids, in translocating them around. So there is still an energetic cost to acquire, to get them out of the soil, to take them up through the roots, to move them around. There's still processing costs. But the difference is that when the plant is absorbing these organic forms, it is also absorbing carbon with that nitrogen. So we have this rebate, what they call the carbon bonus in this example. So you have a rebate, you have embedded carbon that is being absorbed with the nitrogen. And that carbon comes off the cost there. So if we take those gross costs and we add in the bonus or the rebate, that brings our net assimilation costs, our net processing costs down. Okay, so for the plant to use the energetic cost or the carbon cost to use those amino acids is less because they are also have some free embedded carbon that is being absorbed. So when the plant has to process nitrate or ammonium, and remember, it's going to turn those into protein, aminos and proteins, it's going to turn them into organic molecules. That means it's going to stitch carbon into those inorganic forms. We're going to add carbon to that nitrogen to turn them into organic forms. So where does that carbon come from? There's no carbon there. There's no carbon there. So if the plant has to now stitch carbon into that nitrate or into that ammonium and turn it into amino acid, where does the carbon come from? Mhm. Okay, Can I come back to that question? Because I'm going to share a study that compares some different forms of particularly urea with amino acids. But let me come back to that question, and I'll maybe repeat it for the audience at home. Okay, so where does the carbon come from to stitch into those inorganic forms to turn them into organic form? Any guesses? Sugars, which comes from photosynthesis. You got it. It comes from sugars. It comes from photosynthesis. Spot on. Right there is the top. There is the product of photosynthesis, glucose, that sugar molecule. CO2 plus water gives us glucose, gives us the carbon, the energy, the carbon skeleton to then stitch into the inorganic forms to add the carbon. Okay, so don't worry, we're not going to go through this slide in detail. When we are adding inorganic forms of nitrogen into the plant, that photosynthetic carbon, which is the basis of how plants grow, we have to send that carbon down these pathways, down this pathway, to turn that inorganic nitrogen into all of these various amino acids. Okay, and here's a bunch of there's 20 odd essential amino acids, and there they are there. So the more inorganic nitrogen coming in, the more we have to drain that photosynthetic carbon to come down this pathway and be used down here. So what would happen if we fed the plant more amino acids already? What if we were feeding them here? Now what's going to happen to that glucose, to that photosynthetic carbon? It doesn't need to come down this pathway to build amino acids because we're providing amino acids. So we've got all of this carbon / energy available to go elsewhere. Yep, to go elsewhere, whatever. More root production, more tillers, more biomass, more yield. And that's that point. How much biomass per unit of nitrogen can we produce? So we are saving carbon energy to be used for other growth processes. And that, in essence, is what the value of the amino acids and the organic forms provide. Let's look at a couple more examples of the same thing. So here you're looking at a simple little lab experiment. We're growing these plants on these nutrient plates. So they're nutrient-enriched little plates in which the roots kind of spread out and grow on. The first treatment here is glutamine. So this is one of the amino acids. So we're feeding this plant amino acid nitrogen on the left-hand side. The next treatment, they fed the plants nitrate nitrogen, the inorganic nitrate form. So that's what the only form of nitrogen they put was inorganic form. And on the final treatment over here on the third-hand side, you can see that what they've done is they've done a split root system. So it's one plant, but they put half the roots on each half of the tray, and on each half of this little tray, they've put on different nutrients. So on one half contains the organic form, amino acid, and the other half contains that nitrate. Okay, so that third treatment is one plant receiving two different doses, two different types, excuse me, of nitrogen. Now, the nitrogen concentration in all three of these treatments is identical. So it's the same amount of nitrogen, it's just in a different form, just a different quality. Okay, so we're looking at particularly here on root hair formation, root hair production. So we're measuring root hair length on this graph here. So here's our amino treatment, just amino acids, and there is our root hair formation. We can see we have the highest amount of biomass production, the highest amount of root hairs, and that corresponds with this image right here. And if you look closely, you can kind of see there's a lot more root hairs. Amongst these roots, there's a lot more of these fine root hairs running along those roots. It's not super clear, but hopefully you can see it just looks a bit more cloudier, that image, versus this one, which is the treatment that corresponds to this treatment here. Pure nitrate, sole nitrate as the nitrogen source, longer metabolic pathway, greater energy drain to process that nitrate, leaving less energy remaining for biomass production. So there's your biomass production suffering accordingly. Okay, so it drops right down, and you can see the lack of root hair formation. Over here is our third treatment with the one plant root system, but split in half into two different sides, and that's these two sides here. And again, you can see where we provide the organic nitrogen, the amino acid, we have much greater root hair formation as compared to the sole nitrate side. Okay, so there's a nice example. The root length is equivalent to our example of how much production can we achieve per unit of nitrogen. It depends what form of nitrogen you apply. The nitrogen concentration in all of those treatments is identical. Okay, it's the same. But what were we able to do with that nitrogen is very different. Okay, so there's another example. And here, this table is summarizing that, what you just saw, summarizing these numbers. So what we're looking at here is plotting the root mass, how much root mass can we produce to how much nitrogen we took up. Okay, so how much biomass per unit of nitrogen is that question again. The nitrate here is in the red. So the nitrate treatment is in the red, and the blue is the amino treatment. So the first thing we notice is, let's look at the nitrogen uptake. How much nitrogen did we get into the plant? You can see that the red line is above the blue line, meaning we have more nitrate in the plant than we do amino acids. So we were able to get more into the plant actually in the nitrate form. And that's because nitrate is that smaller, simple ion, and it does pass through the roots a bit easier than the amino acid forms, okay? Because if it's small and simple, it can move passively through the roots. So actually, because it's higher on the root uptake axis here, it's above the blue line, so we actually have more nitrate in the plant than we do amino. However, when we plot that to the bottom axis, converting that into root mass, into grams of root mass, into biomass, you can see that despite being lower on the amino form, we are extending further to the right, to your right, right? So we are producing more root biomass because we are extending further this way. So we are producing more mass, root biomass, with less nitrogen. We have less nitrogen in those roots, but we were able to produce more root biomass. More with less.
Okay.
Yeah, there you go.
Jon, kan du ikke lige prøve at snakke i den der mikrofon, nu har jeg lige sat den igen. Ja. Jeg lige har båret nogle ledninger.
Sorry. Just a quick. So you also get free carbon on the blue scale. So if you take the total nitrogen content, it might be the same, like of the root mass. So if the CN ratio is different, you get free carbon. And I also don't understand. Usually the y-axis is the response. So I don't really understand how N uptake is the response from root mass.
It's not, you're right. Of course, we are producing the root mass because of the N uptake. So yeah, it does. We could plot this on a different axis. It wouldn't necessarily change. The graph would look different, but it would still be the same, that we are producing what is the amount of mass we're producing per unit of nitrogen. So, okay, the graph would look a little bit different if we reverse the axes, but as this is just the way the study has laid it out. But because you can see that the blue line is lower, there's less nitrogen in there, but it's producing more biomass. And yes, you're right. The amino form has carbon embedded. This is part of that advantage. That's why that is the mechanism by which we're seeing this difference and seeing the change. So yeah, I'm not sure if I'm exactly answering your question, but yeah. Okay, sure. Okay, sure. So yeah, I get your mechanism. So the extra mass is just the carbon that you added. Sure, I guess that's a plausible scenario, but I don't think, the plants are also growing at the same time. If it was, put it this way, like such a short duration that the plant then doesn't have any time to use that nitrogen to photosynthesize, but give the plant a few more days and it's clearly going to be adding more and more carbon. So yeah, maybe in a very short time duration, yeah, you'd have a point there, but over the course of the experiment, I think that effect would have got diluted out within a few days. Let me give you one more example, and a little bit plugs into maybe the point you're making with one final example. And in this example, the treatment here is using a protein hydrolysate product. So what is a protein hydrolysate? It is a compound. It's a solution or a compound, some product that are a mixture of free amino acids and peptides resulting from digestion, so enzymatic or acid kind of digestion of high protein materials, usually taking high protein kind of waste products, and kind of the idea here is just taking things that are high in protein and upcycling them, so digesting them, breaking them down back into their constituent amino acids. So it's taking anything that's high protein and digesting it, turning it back into things like peptides and amino acids. So just breaking it back down. And you could use anything for this. You could use all sorts of various type products. They could be animal origin, they could be plant origin. So some of the animal origin ones are things like a fish hydrolysate, or taking animal wastes like organs or feather meals or blood, etc. So anything animal waste products, they're very high in protein. And so we can take that high protein material and digest it and break it back down into the aminos. Or you could have a plant origin one, and that would be something like a pulse legume, a seed meal, again, alfalfa, lucerne, anything that's high in protein, plant protein. We could take those materials and digest them back to the constituent amino acids. Okay, so a protein hydrolysate is just taking anything high protein and breaking it down into various mixtures of aminos and peptides and the like. Okay, yeah.
Ability?
There we go. Yeah.
Is it correct that the ability of uptake of NH4 rather than NO3 is very different depending on plant species?
Sure, yes, there would be. And that a little bit taps into that point about pioneer species and later successional plants. Often the early colonizers, the early successional type plants are a bit better nitrate accumulators. And some of the latter successional plants prefer a little bit more in the ammonium form. So yes, there would be differences amongst different plant species. However, actually all plants do have an ability to use all of those different forms. And the main driver of what forms that they use is actually what's available in the environment, what's actually just there. So in this debate, in this discussion about inorganic versus organic, historically, we have said the reason that inorganic forms became the dominant paradigm is because there was evidence in the literature that showed that native plants in wild spaces, natural ecosystems, etc., that those plants do take up some amino acids and organic forms. And when we looked at domesticated plants and agricultural plants, they took up a dominance of inorganic forms. So that built this model that actually the inorganic forms are the more important and more dominant ones. It only seems to be native plants, ecological plants. It was kind of observed in the ecological literature, not the agricultural literature, that amino forms were important. And that was the rationale as to why we went down this pathway. If we actually took our domesticated plants and put them into soils which were not receiving inorganic fertilizers and not being managed organically, they will then adapt and take up what's there. Similarly, if we go and apply inorganic forms to wild native spaces, they will also make use of that. So the big point is, the reason we built that belief that domesticated plants don't predominantly use inorganic forms is because that's how we manage agriculture, and that's the forms that are dominant in supply, and that built that prevailing model. What is the main driver of uptake is simply what is available in the environment, and that supply sends a signal to the plant detects those, and it will actively take those up accordingly. So yes, there are differences between plant species, but actually plants are very adaptable and they will take up what's there. Is there a question?
Just will you share the different protein hydrolysates that are available for organic farmers?
Sure, there would be some out. I mean, I don't know specifically if there are some certified products available, ready-to-use products. I imagine there would be some. I know with organics you have some restriction over the use of animal-based ones. You may have to be a bit careful on some of those. But your plant ones should be fine. And I don't know any commercial products available here, but you can make them yourself also, and through a fermentation process. I don't have a slide in here, but I'll put it in before I share the slides, just with some signposting you to some information on that. You can make your own. You just need to use things like lactic acid bacteria, do a lactic acid ferment. So take the high protein material, grind it down, put it in a drum with some water, add the lactic acid bacteria and molasses, and you're going to ferment that. And so through that fermentation, you're going to let the bacteria produce the enzymes that are going to cut up those proteins and break them down. So you can do it DIY style on farm through a fermentation. And I'll add a link to that. I'm sure there are some commercial products available here. If anyone wants to chime in, does anyone know if there's protein hydrolysate type local products ready to go? Fish hydrolysate you can get? Yeah, okay.
Det kan du bruge til at lave det, kan man sige.
Okay. Okay, so last example. Okay, this one is looking, I hope it will try and answer your question also. So here we're looking at similar root development. So we're looking at a range of various root properties, okay, root branching, root length, root mass, all sorts of root traits that we're measuring in maize. And the three treatments here, you can see black, gray, and white. The black is ammonium, form of nitrogen, okay, so it's inorganic nitrogen. The gray is amino acid, collection of amino acids, and the white one is protein hydrolysate. Okay, so again, the protein hydrolysate is going to be a little bit of a mixture of kind of all of these, depending on how that digestion goes, how fully digested it is. It's going to be kind of a bit of a mixture of these. Might be some bits of bigger protein in there, but a bit more of a mixture. So we're comparing ammonium to amino acids to mixtures of aminos and peptides and the like. And again, the nitrogen concentration on these is the same. It's the same amount of nitrogen that we are applying in all three of the treatments. Now, you can see generally this is a fairly consistent pattern most of the time, whereby the amount of root mass in all these various root traits that we're measuring, the amount of root length, root mass, etc., often has this pattern whereby the lowest production is the ammonium form, and then we go up to the amino form and up to the protein hydrolysate form there. Okay, so sometimes there's not a big difference between these two. Sometimes there is. As you can see, some of these are significant, some of them less so. Now, they did this on two different nutrient gradients, and you can see very clearly this one here is a little bit opens to your question. This one here, you can see we're looking at the total nitrogen in the plant. So here you can see that the inorganic form, which is the black form, has the highest nitrogen, but it's going also down from there. Okay, so we're having the opposite of all these going up in terms of the mass we're producing, but in terms of then the nitrogen concentration in that mass, you can see it's going down. And again, that is the point that's saying that as we produce more biomass, the nitrogen that is there is getting diluted. So we're adding more and more mass so that because the nitrogen concentration is in a percentage here, so the concentration is getting less because we have more biomass. And that's, again, part of that point about more with less is that the nitrogen looks like it's going down. It's the same amount, potentially the same amount that's in there, but we're able to convert more of that into biomass and therefore dilute the nitrogen. Hence why it looks like it's kind of going down there in that example. So yeah, look, I think that, yes, you're right. There's going to be some free carbon coming in, but we'd have to dive into it. But I can't see that amount of carbon is going to be the same difference as all of these treatments here. I think it's more about the additional photosynthesis that it's triggering and the energy saving from that photosynthesis, glucose, that it's funneling into the other growth areas. But yeah, it's a good point. Yeah, we should look into that.
Just to add to your point, the point I just looked it up, and according to the study from before, only the carbon from the glutamine only accounted for 7 to 8% of total biomass carbon in the samples.
Okay.
Yeah, exactly.
Correct.
Yes. But if you had double the biomass with
Yeah.
7 or 8% carbon.
Otherwise, you can see it when you act like you extract the carbon, which fixes carbon, then it's like.
Yes, that's right. Okay. You should come along more often and
Right.
answer quickly double check what the study said, because I can't remember.
I have a more practical question. When you fertilize with hydrolysate, do we know how much of these amino acids from the hydrolysate that actually come into the root? Because I think a lot of these hungry bacteria, they will attack the hydrolysate as fast as they can.
That's right. That's 100%. So that's a really nice question that leads me into this last example I want to give, which is a foliar application of amino acids. And the reason I think this is an important study to share is the exact premise of your question. So amino acids are, as I've outlined, amino acids are very, let's go back to this slide. Amino acids are very metabolically efficient. They save the plant's energy, all of that conversation. But the kroner cost, the dollar cost, your kroner cost, yeah, the dollar cost of those amino acids per unit of nitrogen is very expensive. The kroner cost. The metabolic cost to the plant is much lower, so there's energy to be saved. But the economics of you using amino acids, they are expensive sources of nitrogen because they're quite low analysis, and your cost per unit of nitrogen is very expensive, which opens up this important point of the next study, which is what I want to unpack that dilemma. But because of the very expensive value of these amino acids and protein hydrolysates, I would personally, where possible, recommend them to be used mainly as a foliar application. For the point that is that when you put them onto the leaf, we can guarantee more that we actually get them into the plant. Because when you put them down into the soil, you have a whole lot of hungry other mouths there, the bacteria, the fungi, etc. And yes, they are going to eat those amino acids first, most likely, or certainly a good portion of, a very good portion of. So putting them down into the soil, look, it's not as if that is bad or anything. It's really good. Actually, the bacteria love it, and it will stimulate them and feed them. And that's not wasted. You're still going to get the cycling of that through rhizophagy as those bacteria die and decay. So it's not lost or wasted by any means. You will get it cycled back around. But in terms of just being very targeted and efficient with timing of the use of those high value but metabolically efficient forms of nitrogen, I think it's a bit of a safer strategy to target the foliar, just to make sure we actually get them into the plant. And particularly at key stages of plant development, where you can boost plant growth with particular gains by making sure that you get the nitrogen in at that time. Now, so that said, I've seen some great results with amino acids as a seed treatment. And you get, so there's a gray area. It's on the seed, but the seed is only just germinating. It might be absorbing some of those through the seed, but probably mostly the biology would get at it first. But I've seen some really good studies showing good benefits from a seed treatment. So I'm open to them putting them down into the soil. I think they have a role to play. But if they are costing you a lot, I would lean a bit towards the foliar. Okay, now let's dive into this study. This is a really juicy one. And we're going to kind of wrap up this point about the organic forms of nitrogen. Here we are applying a whey protein hydrolysis. It's made from buffalo milk whey, and they've digested that whey and made that into a whey protein hydrolysate. Okay, so made from whey. Okay, you've got all sorts of opportunities to make protein hydrolysates out of dairy waste, for example. Okay. So what they've done in this study is this is a soil application first of conventional nitrogen. Okay, so we're starting with a soil conventional nitrogen application trial where we're going from soil application 145 kgs per hectare up to 185, up to 215, up to 250. So we have a low to high basal soil conventional synthetic nitrogen application going on the bottom, okay, from low to high doses. And then on top of that, we're going to apply 3 treatments across that nitrogen gradient. We're going to do a control with nothing. We're going to do a foliar urea, and we're going to compare that to a foliar whey protein hydrolysate. Okay, so we've got that low to high soil nitrogen levels, and then we're going to see if we can get an additive benefit from this foliar nitrogen urea versus amino. Now, the general rule with doing low to high soil applications is that foliar sprays, the benefit or the response that you get from a foliar application is usually diminished when you put lots down in the soil. So where you do low soil nitrogen and then do a foliar spray, you'll get a much better response from that than if you do high soil nitrogen and then do a foliar spray. The benefit you get from that foliar spray becomes marginal because you put so much down into the sort of higher dose into the soil. Okay, so that's the normal trend, and that is exactly what we saw here. So here you're looking at the relative advantage on yield. We are looking at the low dose of the soil, basal soil nitrogen, up to one of the higher doses, 215 in this example. And you can see that where we had lower soil fertility, we got a bigger relative response from both of these foliar sprays. Both of them gave a much bigger response than they did at a high soil fertility. We only got a bit more of a smaller marginal bump. Okay, so we saw that normal pattern from both of the 2 foliar sprays. However, if we then compare the urea to the amino form, you can see that the black is the protein hydrolysate, is the amino form. So the black is the amino form and the urea is the white. So under both low and high soil fertility, you can see consistently the protein hydrolysate outperforms the urea. Now, here's the kicker. How much nitrogen did they apply in the urea treatment versus the protein hydrolysate form? The protein hydrolysate has outperformed the urea, and they applied 15 kilos per hectare of actual nitrogen from the urea, and they applied 1 kilo per hectare of actual nitrogen from the protein hydrolysate. So that protein hydrolysate has outperformed the urea at 15 times less the nitrogen concentration. 1 kg per hectare of actual N versus 15. And it is, okay, a marginal improvement. It's a little bit, but still, it is 15 times less nitrogen. So even if it got the same, that would still be outstanding. Even if it was a tad below, that would still be outstanding because it is 15 times less the amount of nitrogen. So that is the, again, metabolic efficiency, the gain that we have through those organic forms. Now, interestingly, that benefit that we saw to yield did not translate through into protein gain, which was very interesting. So, okay, it's marginal, but again, okay, and again, depends how we frame this. Okay, look, the urea did a tiny bit better than the protein hydrolysate in terms of protein gain at low soil nitrogen. And at higher soil nitrogen, again, we saw a slight improvement from the urea there as well. So the urea seemed to did a bit better at building protein, however. This is on wheat, sorry. This is on wheat. Okay, urea did better, but it was applied at 15 times greater concentration. Okay, so I think it is still an impressive result and still illustrates the potential of the metabolic shortcutting and the efficiency gain. So, and again, like I say, I would lean towards using this as a foliar spray because more targeted to get those benefits. So yeah, I have a look at that study if anyone is interested to have a dive into that one. But I think a very, very interesting one to wrap up that section. I would add a small caveat with that study. The study was from Egypt, where for them it's Mediterranean style growing season. So we have hot, dry summers. Okay, so those wheat plants were growing in a very hot and dry summer. And it is no doubt part of the benefit of the protein hydrolysate, that whey protein hydrolysate, is the fact that amino acids are not just metabolically efficient organic forms of nitrogen. They are also molecules that help the plant with all sorts of stresses. And you named this stress, environmental stress. There are amino acids that form specialized little peptides and proteins that help plants deal with those stresses. So this is an example from this paper here, amino acids helping with a whole range of stresses. Sun stress, UV stress, temperature stress, heavy metal stress, salt stress, drought stress, waterlogged anaerobic stresses. All of those environmental stresses, amino acids have been demonstrated to help plants tolerate those environmental stresses. So I'm pointing this out to say that that amino treatment that we gave with that whey protein hydrolysate also provided some stress alleviating compounds that would have helped those plants growing in the Egyptian summer deal with, you know, some of that high temperature stress. So I would argue that some of that benefit we saw in that last study came from the fact that it was aminos and they also help. They are multifunctional, okay? So they also help with stress as well as help to build protein and yield and et cetera, okay? So metabolic regulation involving amino acids is considered the primary strategy for plant protection and survival living under adverse conditions. Okay, so amino acids are key to helping plants reduce stress. So I would add the caveat that I think that's part of that benefit that we saw there as well. Okay, I wanted to quickly share this study. Yep, sure, fire away.
Use this is otherwise use of pretty expensive.
So it depends on the cost of the product. So if it is cost viable for you to apply amino type products down into the soil, yeah, it's fine. And if it's not that aminos down in the soil are bad, they're still very, very good. But if the economics stack up for you to do that, if the crop is high value enough, and so you mentioned it's in a greenhouse context, so if the value of the crop is high enough and the economics dictate that you can use them down in the soil and you're seeing good results, I would happily proceed. But sure, there may be an opportunity for you for some cost savings to do a foliar instead, and you might be able to use a lot less of them. That would be worth exploring. Yeah. Yeah. But the aminos in the soil are not bad by any means. They are still very good to feed the biology and get the benefits through the cycle. It's just that you might be able to do it a bit more cost effectively. Okay, so a little bit linking into this is I wanted to share the study. It plays into a little bit around turning the inorganic forms into organic forms and turning those into the biological forms, into the bacteria, the rhizophagy type cycle, how we can support that process of turning the nutrients into more favorable forms. And this was a study from Australia done on very light sandy soils. Okay, so this is adjacent to the Great Barrier Reef. I'm sure you're all aware we have huge problems at the Barrier Reef now because there's all sorts of nutrient leaching into the reef causing the problems. And I know you have some sandy soils here in Denmark and problems with leaching and things too. So this was done on those sandy soils adjacent to the reef. And what they were doing, where we grow lots of sugarcane, sugarcane's high fertilizing crop, huge amounts of nitrogen use on sandy soils next to the Barrier Reef. I can't see what would go wrong. Can anyone else? So anyway, they're trying to obviously deal with this now, and there's lots of good government programs to try and help farmers with all of this. But so this was a small experiment where they just did something very small and simple. They added molasses to the fertilizer and then applied that to the soil. So we're adding a carbon source, adding some sugar, adding some energy, adding some stimulant for the biology, adding some carbon. And that's all they did, simple as that. So we're comparing N and P fertilizer, and we're comparing that to N and P fertilizer plus molasses. Yeah, we're just going to add molasses to the same fertilizer treatment that and apply that to the soil. So I'll read you a few quotes from this one. The nutrient immobilization, so locking up those nutrients, occurs when the application of readily available carbon, the molasses, when that stimulates the growth of the soil microbes, and it enables them, it encourages them to then eat the fertilizer, to take up luxury amounts of that nitrogen, phosphorus, fertilizer. So by putting molasses, sugar, carbon, energy there with fertilizer, that sugar feeds the bacteria, encourages them to eat the fertilizer and turn it into their bodies. Because remember, bacteria are just like plants, just like animals, just like us. We all require macro and micro minerals to function. So they need those same nutrients. Okay, so when we provide the energy with the nutrients, they will take up the nutrients and grow their bodies, grow their biomass. And that's what they found. So then that immobilized, that locked up nitrogen phosphorus gets put into the bacterial bodies, that this is stored within those microbial cells, and it is released or mineralized at a later date in the plant available form, often released in either inorganic or organic form. But in essence, it's creating an organic slow release fertilizer. It's that N and P from inorganic synthetic source is high availability. But if we just add carbon and we get the bacteria to eat it and store it in their bodies, it creates a slow release mechanism. Okay, we haven't lost those nutrients. We're just temporarily storing them in the body where they can be released later, or they can be released through the rhizophagy cycle. Okay, all those bacteria can be eaten through that rhizophagy cycle. So our results here suggest that molasses amended liquid, that formulation can be considered an enhanced efficiency fertilizer, harnessing this nitrogen and phosphorus immobilizing capacity of those bacteria, soil microbes, with microbial sequestration, ensuring that there is proportionally more fertilizer that remained in that soil for up to 17 weeks. So what they found is where they added molasses, that those nutrients then remained in the soil for a longer time than they did without the molasses, because we lock it up in the biology and are able to store it there and create longer term slow release. Okay, and that slow release is going to be better matched with supply and demand of the plant. Okay, so we can have constant slow release for the whole plant growth cycle. So they also did a nitrate leaching test additionally by adding molasses to nitrate, okay, down in the soil, and they flushed that through some soil columns where they can measure the nitrate leaching out the bottom. So by adding molasses to the nitrate leaching solution at the same concentration of the fertilizer, so 10% fertilizer, 10% molasses into the liquid solution, this stimulated the microbes to, in that soil column, to immobilize 18% of that nitrate within 2 hours and 95% of that nitrate by the 4th day of incubation. Okay, so by adding molasses, we stimulate the bacteria, etc., the biology, to eat that nitrate and store it in their bodies. And once we get nitrate out of nitrate into the bodies, it's no longer going to leach. It's nitrate that's the leachable compound. It's that negatively charged anion that will flush through. As soon as a bacteria eats that, it turns it into what? Into its body, into aminos, into proteins. Bacteria build aminos and proteins too. So that's it. We then get nitrate out of nitrate into the body. Now it's not going to leach through. And bacterial bodies aren't going to leach through so well either. Okay, they're not so leachable like nitrate is either. They're not going to off-gas either under anaerobic conditions where that nitrate can turn into nitrous oxides. So now we just store it in the bodies of the bacteria. Okay, so it's as simple as that. Just add a carbon source to your fertilizers, and that will help to stimulate the bacteria, etc., to feed on those fertilizers and incorporate them into their bodies. Okay, and there are other carbon sources that we could use. Any of these types of things, okay, your humic acids, your fulvic acids, we mentioned the aminos, proteins, seaweed, kelp, molasses, sugars, other plant extracts, you name it. Like any of those are carbon sources that can do a similar job. Okay, so these would be some good strategies with your biodigestate type materials. So when we're kind of reusing some of the digestate materials from biogas, etc., some of those that are very high in fertility. So this point about adding carbon sources, it's equally relevant adding it to nutrients, synthetic nutrients from the bag, but also when we talk about slurry or biogas waste treatments, etc., those may be organic and natural additives, but they are still very high fertility. They still have lots of free nutrients in them. So high doses of slurry high doses of nitrogen is the same as nitrogen from the bag. Okay, there's added carbon there to buffer it to somewhat, but if we're just putting very high doses, which I know we often do in context of slurry because we have too much of it, we put too much of it in, it starts to behave like nitrogen from the bag, etc. So adding some carbon sources to slurry or to biogas treatments, etc., is also going to help to provide carbon energy for the biology to then feed and utilize those nutrients, store those nutrients in their body and prevent them from being lost and leached, etc. So practically speaking, adding carbon, it's a very small, low-hanging fruit, simple step in which we can, which we can help things. Yeah. Yes. Yes, absolutely. So the question here was, through biogas, what are we doing? We are burning off the carbon. We're fermenting off the carbon, so it's going off as methane. So you're losing the carbon through the biogas fermentation process, leaving the concentrated nutrients there. And that, therefore, is the reason that you especially need to add more carbon back to help stabilize that rich nutrient load, as compared to using straight manure or straight slurry on its own, where you've still got more of the carbon embedded in there because you haven't burnt it off through the fermentation. Yeah, 100% correct. So you could argue there's even much greater demand to do that with some of those waste treatments. All right, so I was going to say a few quick things on SAP. We had that question earlier about SAP analysis and tissue analysis. I'm going to do just a few quick comments about this, so we can also move on to our last section. Just to address that question and chat very briefly. Look, in terms of the difference between tissue and SAP analysis and where I stand on this, I'll say this very clearly. Look, I support both. Both have strengths and weaknesses. Okay, SAP analysis is really good. Tissue analysis is really good. SAP analysis has some weaknesses. Tissue analysis has some weaknesses. Okay, they both have strengths and weaknesses. If I had a choice between using neither of them, none, or either one of them, I would choose either one. Okay, either one in my book is better than none, for sure. Now, which is better, tissue or SAP? I think we can have this debate. I think it's still an evolving question. I'm open to both. I use both. I think what farmers are used to and comfortable with using is better than none at all. Okay, so there are some subtle differences. In essence, the SAP analysis, you are measuring the nutrients that are in the plant sap, versus the tissue analysis, you're measuring the whole leaf. Okay, so the analogy with the human body is SAP analysis is like taking a blood analysis. The traditional leaf analysis is like taking my whole body and grinding it up and measuring those nutrients. So, of course, if you take my whole body and grind it up, you're going to get all of my bones, and that's going to give you much higher levels of calcium and magnesium and phosphorus, for example, in the whole plant analysis, in the whole body analysis, as compared to the SAP. Okay, so they are different in that sense, and that presents strengths and weaknesses. Okay, so the SAP analysis is said to be a bit more of a better predictor of nutrient limitations, because the nutrients are coming up into the plant, going through the sap, and then they go to the leaf where they're needed to go. Okay, so if you've got it low in the sap, that's a predictor that you're going to be potentially low in the leaf. The sap analysis is also just a moment in time. It's giving you very much real-time nutrient status at that moment, whereas the leaf analysis does give you a bit more of a cumulative effect, a cumulative measurement. So we might have been absorbing calcium and some of those other nutrients in the past, and now they come up into the leaf and they get embedded into the cell walls where they are locked in. So now they're not in the sap, but it doesn't mean that they're not useful. They're still doing their job, like my calcium is doing its job in the bones. Just because we don't measure it so high in this blood analysis doesn't mean anything. Similarly with the sap analysis, you're picking up only calcium that's in the sap, but you've deposited lots of calcium in the cell walls, and it's still there, and it's still very functional, still very useful, doing lots of good things still. Okay, so that's again a little bit of the subtlety. So with the tissue analysis, the whole leaf analysis, you're picking up of some of that calcium that was previously absorbed, has gone through the sap and now deposited in the leaves. You're picking up on that. So the tissue analysis is giving you a little bit more of that cumulative, historical, overall kind of nutrient supply. And look, there is challenges with sap analysis, sap extraction. So when we say sap, plant sap, there's also gray area. How do we know we're actually extracting nutrients just out of the plant sap? So it's very difficult to get them just out of the sap. It could be coming out of the other cells, ruptured cells, intercellular areas. There's many nutrients that could be leaking out of plant cells that are forming part of the plant sap analysis. It's not exclusively the plant sap. So there's a lab in the US that measures plant sap analysis, but they've changed their terminology and they do say leaf extract. It's a leaf extract measurement. Because if you look at your methodology, some of them take the plants and freeze them and then crush them to extract the sap. But is that really the sap? Well, that's just the cellular contents that after being ruptured and leak out that we're collecting. So one of the weaknesses or gray area over the sap analysis is the point that are we actually measuring the sap or are we just measuring plant juices? And so that is one of the gray areas. One of the other gray areas of the sap analysis is the target ranges, the ideal levels. The ideal levels are not derived perfectly. Okay, so what I mean by that is that the lab in the Netherlands anyway, and others, they will receive samples, and if all of the wheat samples and all of the blackberry samples, et cetera, that they receive, they will put them individually into these big databases, which is a big evolving database that basically averages all of those samples. So we've got this average of what all of the plants are, and we kind of build our target range according to these averages of many hundreds and potentially thousands of samples. And sure, the bigger that data set gets, the more robust it becomes. But you can see the obvious point. If everyone is submitting plants that are low in boron, and so we take the average of everyone's low boron and say that's the ideal range, that's not ideal. So the weakness of the sap analysis is definitely how accurate and how reliable are their target ranges. And the tissue analysis is definitely more robust as compared to that. And that's because the tissue analysis is a more historical method we've been using for many decades, and we have a lot better data sets over what those numbers should be. And that's because a lot of that fundamental work was done over the last many decades using hydroponics, using solution culture, which again, you could say is a weakness too, because does solution culture truly represent what happens in the field? No, so that's also a weakness. But on the plus side, the way we have done that to develop the ideal levels of traditional analysis is to do little hydroponic experiments like this, where we put all of the nutrient solution in, and then from there, we just progressively take one out. We strip nitrogen out. There's no nitrogen. We strip sulfur out. There's the same full nutrient solution without sulfur. Same nutrient solution without potassium or without iron, et cetera, et cetera. So this is a much more robust way. So we do this type of thing and we look at the impacts of that on the plant. Okay, we could take one of these and open it up deeper. So let's just take the first one, nitrogen. Okay, that's no nitrogen. What we could also do, which is what we did do to develop these ideal levels, we then took that complete nutrient solution and we, instead of going no nitrogen, we just dropped it a little bit. And then we dropped a little bit more, and then we dropped a little bit more, and we dropped a little bit more, and a little bit more, and a little bit more. So we did a gradient of nitrogen reduction to the zero. So then we have all these plants growing at this range of nutrient constitutions just for that one particular nutrient of interest. And we find that sweet spot across that whereby the plant biomass and the reproductive capacity is optimized. So meaning we find the right point where the plant completes its life cycle, where it reproduces, where it produces seed, and then we sample those leaves and we measure them and we develop roughly, and it's based on hydroponics, so it's not true representative of the soil, but at least we can say the plant fully expressed itself and reproduced at this amount. Below that, it wasn't able. above that, it also developed other issues, or it was just wasted nutrients, but we're still doing it, but we were able to do it here. So my point is, we've put a lot of decades of science and research into developing those ideal levels, and therefore they are a lot more reliable than a SAP analysis that just averages everybody's results. Okay, now look, I'm a fan of the SAP analysis, don't get me wrong. There are other perks to it. Also, I've alluded to one already. It's definitely better to assess the nitrogen balance. When we tease apart total nitrogen versus being able to measure nitrate, ammonium, and total nitrogen, like that's a huge asset. Like what the SAP analysis does in terms of the nitrogen management, I think is really good. I think it's a big weakness of the tissue analysis in measuring just the total nitrogen. It doesn't give us that full spectrum of the quality of that nitrogen that's in there. So look, that's just like a few comments and a few thoughts on this, and that is certainly quite a big topic about the difference between plant tissue and sap. Those are some of the differences. I'm a fan of them both. I use them both, but they do contain strengths and weaknesses, and it's just about being aware of that. Some farmers have told me that they used traditional leaf analysis for many years and kind of never really got anywhere with it. And when they shifted to SAP analysis, they immediately saw changes and benefits. I've heard many farmers tell me that. So, you know, I'm open-minded to it. I think it's a real useful strategy. But I also know others that have tried SAP analysis and felt like they didn't really get much out of it. And they've just stayed with the traditional analysis, traditional tissue analysis as well. Okay, so I'm actually not bothered necessarily too much about which one you use. I think they both can be useful indicators to help steer your management strategies. You've got to kind of find the one that works best for you. But of course, SAP analysis in recent years has gained a lot of traction. Obviously, a lot of people are using it in recent times. Yeah. Yeah, that's right. So certainly the SAP analysis is a lot more dynamic. It fluctuates a lot through the day. That's right. And that's why, say, for example, the Dutch folks, they do recommend always taking it in the morning at a fairly set time, standard morning time, just to get that consistency. Because yes, through the course of the day, it certainly can change and it'd be a lot more variable. The SAP analysis is just a quick snapshot. It's very dynamic and a lot more variable. The tissue analysis is a bit more consistent.
But this data you get back, yes.
The target range.
Information is about
Yes. Yeah. Yeah. So the comment here is just about that there are these differences with the time of the day, with the sampling, but you're not necessarily picking up on that. You're just going to get a simple recommendation back and how to use that. And yeah, you're right. It's not perfect. It's a guide. And I think this is fine. That's what both the SAP analysis or tissue analysis, both of them do. They both have weaknesses, but what they're doing is just providing a guide. They're narrowing the goalpost, giving you somewhere to aim towards, and they're closing the goalpost could be out there and you could try and kick the ball whichever way. But if we can at least bring it closer, it gives us a bit better target. Neither of them are perfect, but at least they help us line up a bit better and take aim a bit better. Yeah. Okay, let's move on to our last little bit of chat. I'll just say a few quick things on foliar feeding. And it links a little bit to the amino discussion there that we had today. And there's kind of a lot to say here, and perhaps what I would preface this is just, I'm going to kind of scoot through this very quickly and mention just maybe a few key points. But if anyone's interested in some deeper information on foliar feeding, you will find some talks of mine online on YouTube. You can find some where I go, you'll kind of hear this in maybe a bit more detail. And you can kind of fill in some of the gaps. I do have some talks on specifically on foliar feeding online. So yeah, have a look for those. I think just to briefly mention a few key things, like why even bother? I'm sure some of you may not even do foliar feeding, maybe don't even have it being organic farmers. Some of you don't even have a crop sprayer because crop sprayers are only used for spraying pesticides, aren't they? But of course, they can be used for applying stimulants, microbials, organic nutrition, feeds, all sorts of other bio-friendly compounds. They can also be used for. So, and we gave the example today of the amino acids and the benefit of those. And I think that they are more beneficial when targeted directly through the plant. And that you're really going to achieve through a foliar spray. And the benefit of also other benefits to the foliar spray is that when we target the nutrients straight into the leaf, we are bypassing the soil. So when you bypass the soil, that means you're not dealing with some of those lockups and losses. Okay, let's use nitrate leaching as an example. If you apply nitrogen as a foliar, you're not putting that nitrogen into the soil. So how's it going to leach away and cause all the problems that you're having here? And many of you, don't worry, you're not special. Everyone around the world has issues with nitrates, or many people do. So if we avoid the soil and put that straight into the leaf, okay, we avoid that problem. So that also means we can be less damaging on the environment, but it also means we are saving money because we are being more efficient with what we apply. You don't need as much. If you're not wasting as much, if you're not flushing it down the waterways, you don't have to apply so much. So you're saving money. So there's an economic gain to be had equally as well. You can use foliars to improve plant health, yield, quality, protein, some of those examples we touched on, and also improve the nutrient value for ourselves or for our livestock fodder and feed and the like. I think this slide is worth emphasizing. What is the mechanism by which foliars work? And one of the big criticisms of foliars is to say that, you know what, it's not actually that meaningful. You can't apply all of the plant requirements through the leaf, especially for the macronutrients, say like nitrogen or potassium. You can't apply 100% of the crop demands through the leaf. So it's pointless. Don't even bother. You can't meet the demands of the plant through the leaf. And that is true. But that doesn't mean that it's a waste of time technique. No one is saying that you're going to apply 100% of the plant nutrient requirements through the leaf. It's just a tool that you can use on top of soil-based management to get the best out of your plants. So you still need a soil-focused strategy, a soil health-focused long-term strategy, but you can use foliars as one tool to layer on top of that, to top up and to augment and support your soil-based strategy. And so that's important, but we're not trying to apply 100% of the plant mass, although there are some examples of that happening, particularly in a dairy context. I've seen some guys that have transitioned to 100% foliar feeding, and it seems to be working really well. So in some instances, maybe, but generally, fine. It may not necessarily be the goal, but we can still use the foliar very beneficially along with a soil-based strategy. And part of that reason is that what is the mechanism by which foliars help? And the point is that we're not trying to supply 100% demand. We're trying to apply small amounts, small but targeted amounts of nutrients in through the leaf. And when we do that, we're going to use those nutrients to prime photosynthesis, to prime sugar production and all of those other things, the secondary metabolites and the like. And some of those photosynthetic sugars, etc., get sent down to the root system and are released as those root exudates. So foliar sprays help to prime photosynthesis, prime sugar production, prime root exudation. And when we prime root exudation, those high digestible sugars, amino acids, lipids, proteins, organic acids, etc., and carbon compounds, all of those things feed the soil biology. And when you feed the soil biology, the biology will cycle the nutrients from the soil and deliver those back to the plant. So the response that you're seeing from the foliar spray is actually the fact that you stimulated soil biology through this pathway. And that's the kickback you get. It is this nutrients that you are solubilizing from the soil. That's the main mechanism. Now, some of your foliar applied nutrients can also be translocated down to the roots and stimulate root growth, root biomass growth. Okay, and so when you grow a bigger root system, that also means that the plants are able to get more nutrients out of the soil. So really, foliar feeding is just about helping the plant get nutrients from the soil. You're helping the plant feed itself from the soil through bigger root systems and through more root exudation. And that's actually what you're trying to do. And the amount of nutrients that you get back from the soil is way more than the amount that you sprayed on. So we're not trying to feed 100% of the demand. We're just targeted, timely applications, low dose into the leaf to help that plant. That is the mechanism that we're really trying to focus in on. And so therefore, if you do a foliar spray in a tight, compacted soil that has poor soil biology and has not got no good porosity, no good oxygen, no good biology, and so if you do a foliar spray on a tight, compacted soil and you release all of those root exudates down into a soil that is biologically compromised, of course, you're not going to get that return. You're not going to get that kickback. So foliars are not a silver bullet. They work well as a tool within a broader system, within a soil health strategy. You have to be doing that too. Foliars are not going to be your one-trick pony. They just are one tool that can help as part of the bigger system. Now, do plants, yep. Yes. Yes. Yeah, because we'll take some questions at the end. Yeah. So just FYI, it is a natural thing that plants do, is take nutrients up through the leaf. They do this commonly, and there are many examples in nature in which this happens. Sea plants, aquatic ocean plants, they're taking nutrients through the leaf. Plants have been demonstrated to take up nutrients from dust. When dust lands on the leaf, the plants are able to solubilize nutrients from that dust and take them up, meaningful amounts of nutrients from that dust. Animal wastes, be that insect waste, be that bird waste, be that livestock, any manure or urine that lands on the leaf, nutrients can be taken up through that leaf. And even fog, okay, has been shown to supply ammonia gas to plants. So yes, it is a normal and natural thing. Plants can take up nutrients from the leaf. I'm going to just scoot through this so we can kind of move on, because this you can find online, but I'll just mention it here so when you see it, you'll recognize it. We can go through some of the strategies that help to improve the effectiveness of a foliar spray. A good foliar spray is not just about filling the tank with water, throwing in nutrients, and going. Okay, there are important details that you need to design for success. And that is, as I said, you'll find this online. This is the framework I use to help steer that process. It's called FACE, Formulation, Application, Crop, Environment, crop or species. And it's just pointing out that what you put into the spray tank is really important. What you mix in there, the water quality, you want as clean a water as possible, especially if you have hard waters. This is not good for foliar feeding. So you want to use clean water as possible. You can adjust the pH of that water down to slightly acidic, down to around 5.5. You could use vinegar, citric acid for that type of a thing to bring the pH down. So you want to consider what nutrients you add, what chelating agents. So that's where you want to add some of those carbon sources that I mentioned earlier. So these are important details of what goes in the formulation in the spray tank. And I would emphasize water quality, spray pH, and carbon chelating agents. And again, you'll see some information of that online. Of course, how you apply it, you want to make sure you reach the leaf. Don't apply too much pressure and bounce the droplets off. Don't use not enough pressure and fall to the ground and not reach the target. So just make sure you coat the leaf surface with the spray. Okay, appropriate nozzle size, not too small, not too big, just nice coating over that leaf surface. Different crops or different plant species have more or less benefit or response to foliar spray. So plants that are very waxy in leaves generally have a bit of a they're not as responsive. So things like the brassicas or the onion family, those very kind of harsh, waxy, repellent type leaves. But that said, it can be somewhat overcome with the inclusion of things like wetter stickers, spreaders, just to make sure that foliar spray does, when it sticks to those waxy leaves, that it spreads out and doesn't just bead. I'm sure you've all been out and seen a brassica crop in the morning and it's all covered in beaded droplets. So we want to spread those out and get them sticking onto the leaf. And that's where some of those additives, wetter sticker type things can be advantageous. And then the key point on the environment, is just to say that the humidity is a really important driver as well. So you want to be quite high humid. The magic number here is about 70-odd%. So above 70% humidity is kind of optimum for foliar spray. But I would encourage you all to check out Delta T. And again, you'll find some info on this. You can punch in Delta T online, and it plots the humidity with temperature. And that also just gives you a chart of a good time to foliar spray. And basically, you want to be anywhere in that green zone there. So look up your current temperature, look at your current humidity, and you can plot those two. And if you're in that, you can type in Delta T into Google Images. You'll come up with oodles of these. If you're anywhere in that green zone, generally, you're good to do a foliar spray. You shouldn't do it on a really hot and a really dry day. Okay, the plants are going to be closed stomata, protecting themselves from that heat. They're not going to be open and porous and take in that foliar spray. Okay, so use Delta T, and the timing of your application is also very important. The general rule here would be that earlier mornings, later afternoons, and in the evenings is the better time when temperatures are a little cooler and humidity is up. That would be the main takeaway message. And I'll say just a couple of quick things on biostimulants, just so we've ticked all the boxes here. Again, you'll find some things online about this. But biostimulants are also useful additives that could be added to a foliar spray, and they can also help to make foliar nutrients work better, but they can also stimulate plants in a non-nutritional means. And that's what a biostimulant is. A biostimulant is not a biopesticide, meaning a pesticide of natural origin, a biological pesticide. It's not a biofertilizer, meaning a microbial inoculant, but it is a compound that can stimulate plant growth in non-fertilizing or non-plant protective means. So it's not protecting the plant, it's not fertilizing the plant, but it can stimulate the plant through these non-pesticidal or non-nutritional means. And that's why they are useful additives that can work well with your plant nutrition approach. And to give you just the examples of what some of the common and main biostimulants are, you've got the humic substances, humic and fulvic acids, you've got amino acids and protein hydrolysase, so we touched on those today. Not only are amino acids organic sources of nitrogen and valuable because of that reason, but they also do have these biostimulant type effects with them as well. You've also got algae, so that's your seaweed, kelp type extracts are very common. You've got a range of botanical or plant type extracts, and there's many different things that you could use, various leaf, the old nettle tea, for example, comfrey teas, weed teas. You've got essential oils, plant-based compounds, and there are many thousands of them, which can also be used. You've got microbial inoculants, inorganic compounds, etc., etc. There's a whole range of them. The challenge with biostimulants is that they are a very, very diverse array of compounds, and they have multiple compounds, multiple mode of actions. And that is what makes them a little bit tricky to navigate, which is that when we say, oh, I used biostimulants and they didn't work, or I used biostimulants and they worked really good, that's not very useful. We need to be much more specific about what biostimulant. Biostimulant is a category. What did you actually apply? Was it kelp? Was it fish? Was it amino? Was it etc., etc.? Okay, so we have to be more specific. There are many different types, thousands of different biostimulants. I would just encourage you to be a bit more specific about which ones. And my last point about them is to say that where we also see some fair enough, good, healthy skepticism about biostimulants, and fair enough, there are many, many products on the market now. And look, I'm sure there are some really good ones, and I'm sure there's some probably not very good ones. But I would just emphasize that for some of the biostimulants, it's important to understand the mode of action, because there's a lot of healthy skepticism because some of them are applied at a rate of 10 mils per hectare or 50 mils per hectare. And some folks might think, how on earth is 10 mils per hectare doing anything? Okay, it is an insignificant amount of this material. But for your understanding, not all, but for some of the biostimulants, the way in which they work, the way in which they can be beneficial, is that actually they are just a signaling molecule, a communication chemical. They can trigger genetic expression in the plant, and therefore you only need the tiniest amount of them. For example, in the plant cells, it has all of these receptors, and you only need the tiniest amount of the compound to trigger the cell receptor. And if that receptor gets triggered, it sends a message down into the nucleus to switch on genetic expression, to upregulate or to change genetic expression. And that is what gives the growth response. And it could be all sorts of changes in genetic expression. It might be helping with stress. It might be helping with pest or disease. It could be doing all sorts of things. But the point here is to say that all we're doing with the biostimulant is unlocking the potential of the plant. The plant's actually doing the work. It's the one that's already got the genes, etc. All this was a low dose to trigger the expression of the plant's own genetics. And that's why sometimes some of these biostimulants are used in incredibly low doses. So just for your understanding, it's not necessarily all smoke and mirrors. There are plausible mechanisms by in which and how they work. But look, I acknowledge there's many different kind of products and things out there these days. So take recommendations from each other, find products that work for you, share, network, collaborate, and share that information with each other to help kind of chart your path forward there. So yeah, in summary, look, still an emerging technology. I think we do have still a bit of a way to go, but there are more and more products coming out. Humic acids and seaweeds, if you look into the academic literature, probably those are the two categories that are the most well studied, I would say, have the kind of more robust evidence behind them. And because they're different compounds, look, there are many different benefits in which they can bring. So, okay, I know we scooted a little bit through that last little section there, but I feel like we covered some of the key areas of kind of foliar feeding and just for your understanding a bit on the biostimulants. But if anyone wants to find out more, just YouTube me with foliar and some of these words and you'll find some information. Okay,
Yes.
thank you very much. Sorry about that. Yeah.
So we are already on overtime. But yeah, is there one last question? Yes.
I was wondering if you had, in the stat analysis, do you have any comment on the use of EC or the pH?
Sure. I use them as a guide. I would say I don't lean too heavily on either of those two. I look much more at the actual nutrient levels. But look, yes, I think, of course, the pH tells us a bit about that acid-alkaline balance, and the EC certainly, if the EC is low, that is potentially telling you that there's just not enough nutrient supply overall. So however you could kind of increase nutrient supply, nutrient cycling from the soil to try and bring that up, I think they're both a piece of the puzzle. I think I would lean and say that EC might have a touch more value, but I'll be honest and say I more so look at the nutrient levels and target those. Yeah, much more so.
We have one last one, short one.
Yes. I was just thinking about sort of, you talked about the chemistry stuff, like we should, in Denmark, we should not use the pesticides, like trying to avoid that. But what about other like pollutant stuff? Like right now, there's a lot about microplastic. Like how much do we know so far on how the different pollution has an effect on these things and the plants?
Yeah, good question. There's a couple of reviews that I've seen looking at microplastics in soil, and yeah, unfortunately, it's common there too. Like it's certainly not just the ocean, and as we now realize, they're also all in our bodies. Microplastics, unfortunately, are everywhere. So, look, there are some papers that I've seen outlining, and there are a few different exposure routes, and the kind of the biosolids and sewage sludge and things is definitely one of the major contributors. So using sewage on land, on soils, is one of the major pathways by which we put plastics into the soil. So that is definitely, yeah, the major source, which we have to be mindful of as well, for sure. But the effects of that on the soil biology and things, I think, is still, yeah, emerging. Yeah, I think we all know this problem has become a lot more perverse and more widespread than we maybe had previously thought. I kind of thought it was a problem for the oceans, but actually it's everywhere now. So yeah, it's still a evolving topic, that one. Yeah, unfortunately.
All right. Thank you very much, Joel. Thank you for coming.
Pleasure. Thank you for having me.
Thank you to you guys who came here today, even though it snowed a lot. Thank you for you guys coming on online. Perfect. Thank you to my colleagues at Organic Denmark for making that possible, and thank you to Innovation Center for Organic Agriculture, Farming, sorry. Yes, yes. I'm gonna switch into Danish now.
Okay. Yeah, no worries.
Der er en lille pose til jer, der ligger deroppe. Der er lidt forskelligt materiale i, der er en croissant. Så ligger der sådan et kort, hvor der lige er sådan en QR-kode, man lige kan scanne. Og der er et par spørgsmål, som vi vil håbe, at I vil besvare, om hvordan det har været at være her i dag. Så kan vi måske